Hi everyone. Thanks. I'd like to thank Gabi and Lars that are actually the two people that brought me on board, not just for the data analysis, but physically on board also. And I have to thank also the 260 people that worked around Tara, because it's with all the interaction that we had over the years that we finally came out with this work and this analysis that I'm going to present. So I'm going to talk about the carbon pump, the biological carbon pump, and so I'm going to just say a little bit something about it. Okay, so this is a global view, basically, of the carbon pump, and what I'm going to focus on is this component here, the euphosic zone, where you interactions. And basically all the bugs here that you have, they play together to create particles that are going to sink. And this is the part of the carbon that is pumped basically by this organism and sent to the depths for sequestration. So it's a very simplistic view of the carbon pump. And over the years, for some of you that know maybe the GGOS program, a lot of studies have been focusing on this and trying to make it very simplistic to be able to understand the link between the ecosystem and the carbon export here. And so what people have been doing, it's really to try to simplify it to the maximum to be able to understand it. And the view that we usually have is just that when you go from poor area, oligotrophic region to eutrophic region, you have a change in the planktonic community that go from small cells to large cells, and that change is going to impact the export of carbon. But that's very simple, and Tara can bring a lot of more information into the picture now. And for exports, that is basically just that arrow here. People have been using sediment trap. They're pretty easy to deploy. Basically, you put a trap, that is a physical trap in the water column, and you capture whatever is sinking through a layer. And so they put sediment trap at different depths, they measure the carbon flux, and then they can calculate an export of carbon or the remineralization, meaning that the remineralization is the way the carbon is consumed by whatever is in the water, and get to the sequestration. So it's very easy to use, but the other way, it's that with this kind of instrument, you have very low spatial resolution and temporal resolution of the carbon export. So basically, it's hard to relate the dynamic of the ocean to whatever is happening down below. And so the main picture of it is that when you go from poor area to rich area, you increase the flux, but at the same time, you decrease the efficiency of this flux to get to the bottom. So meaning that when you have a lot of food escaping the euphosic zone, the top layer of the ocean, a lot of organisms are there to feed on it, and so they decrease very efficiently the way the flux get to the depths, to different depths. And this is because different type of mechanisms that are using that flux. Basically, in oligotrophic region, it might be mostly dominated by microbial degradation. That is a slow process that doesn't affect a lot what is sinking, compared to eutrophic area where you have zooplankton, for example, feeding on the stuff that are sinking and degrading the flux very efficiently. But that's basically the GGOS picture. And here with Tara, what we try to do is to get another picture of it. And people usually don't understand that very well because of the low resolution of sediment trap. In the deep ocean, you have not a lot of information. Just a quick example from the literature. If you take whatever has been published on the moon over the past 100 years and whatever is published on the deep ocean, you have a factor of 30 between the two. So basically, we know 30 times more stuff on the moon than we know on the deep ocean. And this is why basically we don't get it yet. So when you calculate carbon budgets, whatever is sinking doesn't match whatever is needed in the deep ocean. And that's a big black box for now. So what we've been trying to do with Tara is to replace, basically to use the omics with the classical biogeochemistry view of the ocean and to put that into the context of the biological pump. So basically it's to replace this box, the bugs, with genes, and the flux with a new way to look at it, basically using optical instruments like the UVP. This kind of instrument doesn't give you a real flux, but at least it gives you a proxy of the flux. And just for example, if you get back to the historical data from the GCOS programs, using this optical proxy during the Tara tract, we got as much data on flux that has been collected for the last 30 years during the GCOS program. So that's what we did. Basically, we got the big hairy ball that is the interactome from Irun that presented earlier. So that's the biology. And we put it in front of the biogeochemistry of the ocean, that is the carbon flux around the Tara track. So that's the two data sets, but the big issue here is how to make the link between the two, because this is really complex. This is not simple, but when you have to compare it and when you try to get some understanding of the biology and how this is going to impact the carbon export, it's even more complex. So we try to develop ways to look at it by simplifying the data set, but not losing the complexity that is in it. And so first, we had to extract some information from the interactome. So that's what we did by using a system biology approach, the WGCNA here, that basically look at the structure of the network and correlate the structure to environmental parameters. So here are the different structures, the different part of the subnetwork that correlate to different environmental parameters, and here is the carbon export. And that's just for the eukaryotic, so basically the paper presented by Colomban before. And the first surprising result is that when you look at this heat map here, there is only one section of the subnetwork that was significantly associated to carbon export for this community. So this is interesting and very useful because then you can extract only a subpart of it and try to make sense of this. That is much easier than making sense of the full thing. So when you blow this part away and you try to look at what is inside, basically you have this community here. And we try to extract features of the subnetwork to try to make sense of it. So first, it's the correlation of each taxa into that subnetwork to the carbon exports. And the second feature is the centrality of this species into the subnetwork, meaning that species that are here are highly correlated to carbon export, and at the same time, they're very at the center of the network, so they maintain the stability of it. And if you have a species here that is highly correlated to carbon export but is not central, if you take it out, basically the subnetwork is not affected. And we extracted a third parameter of this, basically by using a multilinear regression kind of model. And basically it tells you that among all of this, if you only take few of these species, you can actually predict the carbon export with a certain confidence. And so just by using few species here, we were able to predict the carbon export as about 75%, just by using the eukaryotic component. And what is actually interesting here is that these species that pop out are not the one that are, the one that you think first when you think about carbon export. You would think about copepods, for example, or diatoms, because people, when they work on diatoms, they usually try to work on diatom to try to relate that to carbon export because it's the main primary producer. But if you look at the species that come out, diatom are there, but they're not as important as you would think into that network. Some are, for example, the radiolarian that Fabrice is going to talk about. And so we did that also because doing it for the prokaryotes is fine, but we know already a lot about prokaryotes, but we don't know much about the eukaryote, but we don't know much about the prokaryotes. So we did the same analysis for the prokaryotes, and we had a different community that came out with Synechococcus, for example, being one of the central key species related to carbon export. And with that subnetwork, you can predict about 60% of the variation of the export. And the final very surprising result that was on the viruses, because we did exactly the same analysis, and the species, the viruses that came out were the Synechococcus phage. This is completely independent from the previous analysis, and even though the species that came out are somehow related to the species that were important in the prokaryotic component. And more interesting is that 90% of the variation of the carbon export can be predicted by only few of these viruses. And what we did at the end is, so these three results are based on the three science papers from Jen, from Colomban, and Chini. And finally, we tried to rebuild the network that was the full network using all the components that is correlated to carbon export. And so for that, we used Hiruntalk with the Haribo, except that we extracted the link, the interactions basically in that network, and plotted it against the species that were selected for carbon export. And this is what you get. And what is interesting is, for example, Synechococcus here, that wasn't suspected to be important, but here you understand a little bit why, because it's very central and a lot of things are connected to it. And the last thing we did is we look also at the functions, the functional network of the prokaryotes. And I'm not going to go into the detail, but we did the same analysis extracting few functions that are important for carbon exports. But what is actually interesting is that into these functions, more than 60% are unknown. So there is a lot to do on this unknown function because they're there, they're probably important for carbon export, but don't even know what they are. And so this is just to put that into the perspective of a few things that are coming out, is because now you have environmental sample processors that are coming out on the market. I mean, not yet on the market, but that are available. And because we are able to target few genes, you can think about putting that on this kind of sensor to be able to target few processes into the environment. And I just want to finish with few take-home messages, is that basically what we did is to develop a graph-based approach that is robust and useful to decipher global oceanomics data set, meaning that you can use these to extract in a few information of the omics to be able to link that to some biogeochemical parameters, for example. That we extract few the sub-networks that are related to carbon export for each of the life kingdoms. And basically related to carbon export, what was expected is that few species that were not thought to be important for carbon export came out of this analysis. And again, radiolarian was one of them. Fabrice is going to talk about it. And that we were able to predict carbon exports with viruses up to 90% of the variability of carbon export. That was really not expected. And some engineering outreach, it's that because of this analysis, now we can target few genes to put that on environmental sensor to do some monitoring. And we hope to be able to build or reconstruct metabolic network associated to this gene set network. And with that, I'll answer questions.